Good morning. Thank you for joining us today for MDIC's webinar on patient values and p-values, exploring a new method for developing patient-centered clinical trials. My name is Stephanie Christopher. I am the Program Director for MDIC's Science of Patient Input, and I'm delighted today to be able to introduce our speakers for today's webinar. We have three speakers joining us today to talk about a recent paper that they've published and how that's informing some of MDIC's work in the Science of Patient Input. Somesh Chowdhury from the Laboratory for Financial Engineering and Department of Electrical Engineering and Computer Science at MIT. Martin Ho is the Associate Director for Quantitative Innovation at CDRH. And Andrew Lowe is the Charles E. and Susan T. Harris Professor of Finance and the Director of the Laboratory for Financial Engineering at MIT. We are pleased to have our speakers here with us today, and they have reserved the last 10 to 15 minutes of our presentation today for questions. If you have any questions, please type those into the chat box, and we will be able to read those off when we get to the Q&A portion of our program. So with that, I'll turn it over to Dr. Lowe. Dr. Lowe, you should have control of the keyboard.
Well, great. Thanks very much, Stephanie. I'd like to begin, first of all, by thanking Stephanie and MDIC for allowing us to speak with all of you today and present some of our recent research. And I want to thank our collaborators, Martin Ho, Telba Irony, Murray Sheldon, and of course, Somesh Chowdhury. You'll be hearing from Martin and Sho a little bit later on. And we also want to thank the FDA and our partners at RGI and the Michael J. Fox Foundation for some of the work that we're going to be telling you about. The title of our presentation is Patient-Centered Clinical Trials. This is certainly not a new concept to FDA or to the individuals on this call, but what we want to do is to talk specifically about how we are proposing to take into account patient preferences and the costs and benefits of various different therapeutic innovations that are going on. So I'd like to start with a little bit about the basic thrust behind a clinical trial and the statistical inference that underlies it. In thinking about whether or not a therapy is effective or ineffective, we have to do tests using patients and making statistical inferences. And in the end, the two decisions that the FDA and other regulatory bodies are left with is either to approve or not to approve a given therapy. So if you take a look at this chart on this slide, there are two possibilities for whether the therapy is effective or ineffective, and two possible actions, either approve or reject. And the standard clinical trial framework is to try to reduce the mistakes that you're going to be making, often called type 1 and type 2 error. Type 1 error is a situation where you're approving an ineffective therapy, and given that there are side effects and potential adverse consequences, you want to be very careful not to engage in that kind of error. The typical approach is to set the type 1 error at something like 2.5 or 5%, to look for therapies that have p-values that are below that threshold. But there's a second kind of error that you can make, which is type 2 error. That's the mistake of not approving an effective therapy. And typically, we worry about that kind of error as well. Now, it turns out that we can actually drive these errors down to zero individually. Very simple. For example, if I never ever want to approve an ineffective therapy, if I want the type 1 error to be zero, the answer is very simple. Reject all therapies. Now, it's not a very satisfying response for patients, but that is going to control the type 1 error to zero. Similarly, if you want the type 2 error to be zero, you can accept all therapies. And that's obviously not acceptable either. So clearly, there's a trade-off between type 1 and type 2 error, but we don't really talk about that trade-off explicitly, although all of us are thinking about it implicitly. And our approach is to try to figure out how to do this in a systematic way. In other words, how to calculate the particular trade-off between type 1 and type 2 error in order to do the best for patients. Now, this is not a new issue. Obviously, the FDA has already issued guidance back in 2012 that patient tolerance for risk is actually an important factor to consider. For example, instead of using a 5% p-value, patients might very well be willing to take a higher risk of a type 1 error. In other words, they may be willing to take a higher risk of a false positive if they're dealing with a particular therapy that is meant to treat a disease that currently has no existing therapies and where we're talking about a mortal illness, something like, say, glioblastoma. And in the 21st Century Cures Bill actually now requires the FDA to incorporate patient preferences, and more than that, it also requires the FDA to report on a regular basis of how it's doing so. So that really is the motivation behind what we're trying to do. In this webinar, we're going to describe to you our approach, which is a well-known technique called Bayesian decision analysis, and we'll provide a specific example using data from a number of cancer clinical trials, and then go into some of the applications on the devices side, and then ultimately conclude by talking a bit about what we might be doing prospectively with a Parkinson's disease study. So let me very quickly just outline the basic structure. I know that a number of you on the call have very strong biostatistics backgrounds, so I apologize in advance for going over things you already know, but we would like to make sure that everybody is on the same page, and so I'm going to try to describe things at a relatively high level, and for the details, the technical details, you can see the two published papers that we have, and are happy to talk in more detail during the Q&A. So the basic idea behind Bayesian decision analysis is to do what I mentioned at the very first slide. That is to figure out what the trade-off should be between type 1 and type 2 error, recognizing that there are costs to both of those kinds of mistakes. And so the typical approach that a Bayesian decision analyst would take is to try to minimize the average cost of those two mistakes. If you think about minimizing the expected cost and calculating the probability of a type 1 versus type 2 error, what we'd like to do is to reduce that combined cost when you weight them by the appropriate probabilities. So that's what's shown on this slide here. Once you make assumptions for what the cost of a type 1 error is, C1, and what the cost of a type 2 error is, C2, you can actually calculate what the appropriate weighting would be to calculate the expected cost that you get across various different choices for your type 1 versus type 2 error. In other words, what you'd like to be able to do is to pick an appropriate threshold of type 1 error as well as an appropriate sample size to get the type 2 error that you want in order to minimize the average cost of those two mistakes when you weight them by their respective probabilities. So the BDA optimal decision minimizes that average cost. And the surface that we display in this graph here is a depiction of how you find that minimum. Now, we actually take this framework and have applied it using data from actual clinical trials from the Alliance trials in the National Cancer Institute. This was published in JAMA Oncology just earlier this year. And here's an illustration of what we found. So using data for the burden of disease for patients, in other words, what are the quality-adjusted years of life lost and what the impact would be on patients in trials, as well as the potential for toxic side effects and so on in patients out of trials, what we did was to calculate the optimal type 1 error that we might get, as well as the optimal sample sizes using our framework, and compare that to what the appropriate size and sample size was for the Alliance clinical trials. So in this graph, in blue, you see the sample sizes that have been used in the Alliance trials for various different kinds of cancers. And in green, you see the sizes that have been developed from our BDA optimality procedure, and the red dots indicate the type 1 error that we're getting in order to minimize the expected cost. So let me just give you a couple of examples from the slide. If we turn to something like glioblastoma, this is a disease that is obviously terminal. The 5-year survival rate is on the order of 5% for patients diagnosed with glioblastoma. And currently, there is no existing therapy that will do anything to really treat the disease. So the Alliance trial obviously uses a standard 5% type 1 error, and the sample size that it's getting is something on the order of about 400 patients. that obviously gives you the appropriate type 2 error that you're going to get. Well, if you look at the BDA optimal parameters, what you're getting is a much, much smaller sample for the type 2 error. In other words, it's such a terrible disease that we should be able to come up with an answer of whether or not we approve or reject a drug with a relatively small sample size. And more importantly, the type 1 error is nearly 50%. Now, I know what you're thinking. That sounds insane to have almost half of these therapies approved. But remember, we're talking about a disease that is fatal, and there are no existing therapies. So clearly, a 5% type 1 error is way too conservative. If you talk to patients who are going to die anyway, their response is, oh, we'd like to try whatever we can. And obviously, there are concerns about how this gets implemented, so I'll talk a bit about that at the very end when we conclude. But at least this is what the analytics are telling us that we should use for a type 1 error for these kind of terrible diseases. At the opposite end, let me ask you to turn to the very right-hand set of bars, prostate cancer. Prostate cancer is a situation where you can see that the type 1 error produced by our optimization procedure is actually lower than the typical 5% that's used in standard practice. And the reason for this is pretty simple. First of all, prostate cancer is not terminal. In some cases it is, but in many cases people can live with it for quite a few years. There are a number of existing therapies for it, so the burden of disease is not nearly as heavy as it is for glioblastoma. And more importantly, the number of patients that are affected by prostate cancer is huge. So if you produce a therapy that is ineffective and can have toxic side effects, you're actually hurting a much, much larger population than the population of people with glioblastoma. So in that case, when you run the optimization, what our procedure is telling us is that for prostate cancer, where people can live with the disease, where there are many other therapies, and where you're going to be affecting a very, very large number of patients, you have to be very careful about approving a drug. So you want to be more conservative than the typical 5%. So anyway, this is a bit of an overview about how it applies. And you can see the details on this slide if you're interested. What we have to acknowledge, of course, is that there are many qualifications about how you take these ideas and apply them. For example, how do you choose the parameters? How do you calculate the burden of disease? Our view is that you really need to have a group of stakeholders that would provide input to the FDA, but ultimately the FDA should collect all of this information and incorporate it together in some fixed set of parameters that they publicize, and those parameters can change over time as patient preferences change, as technologies change, as the burden of disease changes. Also, we have to be careful about whose preferences are reflected. We need to incorporate the various different aspects of patient preferences because not all patients approach it the same way. And finally, we have to be very careful if we are going to increase the number of therapies that get approved by allowing p-values higher than 5%, we have to be very careful in post-approval marketing and monitoring as these drugs get marketed. It may be the case that we want to consider some kind of temporary approval, a third category where we allow for very, very difficult to treat diseases like glioblastoma a period of 2 or 3 years where we can allow the drug to be used on an experimental basis, and either that drug gets approved to a regular status or it gets taken off the market after that 2 or 3 year period. In any case, there are many issues that need to be worked through, and we're honored to be working with the FDA as well as the Michael J. Fox Foundation and RGI to try to measure patient preferences and deal with some of the practicalities. And I'm going to turn it over now to Martin Ho of the FDA so he can describe to you some of the work that we've been doing with him and his group in taking these ideas and trying to implement them and dealing with some of the practical issues. So with that, let me turn it over to Martin.
Thank you, Professor Lowe. So in the cancer example, we saw one approach to quantify the relative burden of type 1 versus type 2 error rates. And next, we will use a different approach to measure the patient's the relative burden of the disease to the patient, which is we directly ask patients the relative weight of burdens to them. And yes, okay. For that purpose, we are going to use the CDRH, which is the Center for Devices and Radiological Health, the patient preference study for weight loss devices as an example to describe what exactly we measured and how we measure it. To understand the benefit risk trade-off context, let's take a look at this graph first. Here, the x-axis and y-axis represent the average benefit and risk of a weight loss treatment, respectively. Specifically, the benefit here is defined as the amount of average weight loss. Here, we can see that the diet and exercise is always the first-line treatment suggested by the providers, although it is extremely safe, but the benefit in general is minimal. Compared to diet and exercise, medications can deliver greater weight loss on average, but also associated with additional risk. Next, we are going into the more invasive treatment options, such as gastric banding and gastric bypass procedures. They definitely can deliver more impressive weight loss benefit. However, they also associate with a significantly greater risk. So suppose a new device is under review for pre-market approval, and the new device is definitely safer than the gastric banding. However, there is the benefit of the new device is not as big as the gastric banding devices. So how can we say that this kind of how can we say that is whether the new device benefit risk trade-off is acceptable to patients? The CDRH patient preference study for weight loss devices was designed to answer exactly this question. Let's take a look at the study design of the survey. So the study is a survey jointly developed by the CDRH and RTI Health Solutions. It includes about 650 obese subjects recruited from an internet panel. The survey provided tutorial and quiz to these subjects to inform them about the benefits and risks of weight loss devices, as well as the probability concepts. The survey was administered through the internet. It oversampled subjects who underwent prior weight loss reduction procedure to capture the before and after experiences. So how did the survey capture the benefit risk trade-off preferences of patients? It used a scientific survey format called discrete choice experiment. This term corresponds to a specific format of questions that allow us to elicit the patient's preferences when they are making trade-off. Specifically, the respondents will evaluate choices between pairs of hypothetical weight loss device treatments. And each treatment is defined by its attributes, including the surgical procedure for device implantation. And each attribute has a set of levels or values. For example, the amount of weight loss, the duration of weight loss, and the mortality risk. And the resulting pattern of choices made by the patient will reveal their preferences. For example, using this quantitative format, we can estimate and make such a following statement. The patients would be willing to tolerate 2 or more months of mild state side effects to achieve an additional weight loss of 25 pounds. Now, this is an example of the questions that posed to our survey respondent. To those of you who subscribe to Consumer Report, this type of product comparison would be looking very familiar to you. And here, each device is characterized by 8 different attributes. They are the type of operations, recommended diet restrictions, the amount of weight loss on average, the duration of weight loss on average, the average reduction in the dose of prescription drugs or diabetes as a result of low weight, and on average, how long the mild to moderate side effect would last, and what is the chances of a side effect that would requiring hospitalization, meaning the side effects that are more serious. And last but not least is the chance of dying from getting the weight loss devices. And after comparing these 2 devices, the patient or the respondents would be asked to make a choice, which is which weight loss device do you think is better for people like you? The questions being posed this way is to try to remove the social stigma on obese patients, so that they can more rationally consider those choices. Here we can see that the device B is chosen, is more preferred over the device A. And we can see that the device B delivers 60 pounds of weight loss on average compared to the 30 pounds by device A. And it seems like the device A has much shorter duration of the mild to moderate side effect than device B. However, this patient judged that the greater amount of weight loss is more important than tolerating additional 11 more months of more mild to moderate side effects. Next, I would like to talk in details about individual attributes and their levels. For the type of operations, we will be presenting 3 different options for different hypothetical devices that include endoscopic, laparoscopic, and open surgery. For diet restrictions, we propose 3 different types: eat a quarter cup at a time, wait 4 hours between meals, and can't eat hard to digest food or their favorite food. The average weight loss range from 5% of total body weight to 40% of total body weight. The duration lasts from 6 months to 5 years. The comorbidity improvement range from none to eliminate the risk or the current dosage of the medication. As for the duration of the mild to moderate side effects, it ranges from none to 5 years. The chance of serious side effects requiring hospitalization from none to 5% hospitalization for surgery. Lastly, the chance of dying from getting weight loss ranges from none to 10%. So these graphs represent the study results, which are the point estimate and their standard error of each attribute preference score. Here, the higher the preference score is, the more valuable of that attribute level to an average patient. For example, here 0% mortality risk associated with 10 units of preference score compared to 0 preference score of mortality risk of 5%. So the better outcomes, we would have a larger or greater preference scores. Next, we are going to talk about some categorical attributes. Here we are talking about the diet restrictions. We see that the patients are indifferent about whether they need to wait for 4 hours or eat a quarter cup at a time in terms of the recommended diet restrictions that comes with the devices. However, they are very serious about not being able to eat their favorite food, which is specified as a no sweet or hard to digest food. which was consistent with our general observation during our pretest of the survey instrument, that the patient expressed extreme preference of still being able to eat burger or pizza. So next, I also want to tell you that these different preference scores not only being useful for comparing different levels within an attribute, they can also be used to compare the relative importance of different attributes. Here we see that the range of the preference score for mortality risk is from 0 to 10. For weight loss, the amount that is from 0 to about 4.3, and for weight loss duration is similar magnitude of range. And these range indicated that these three attributes are the most important attributes among the 8 that we are studying. And the side effect, the side to moderate side effect is the least important attribute, which is also consistent with our pretest exercise when we are testing out the instrument, meaning that the patients, when they are receiving medical devices, they already expected there would be some minor side effects they would need to endure in order to achieve weight loss. Next, I'm going to turn it over to Shou to discuss how we're going to use this patient preference information to make clinical trial design more patient-centric.
Great, thanks, Martin. Just give me a second to get control here. Okay, so my name's Shamesh. I am a PhD student in electrical engineering and computer science at MIT. And the topic that I'm going to cover today is how can we incorporate these patient preference scores that Martin was describing into our Bayesian decision analysis framework to design patient-centered clinical trials. And so the details of what I'm going to present are all available in our recently published article, Patient-Centered Clinical Trials, which is available in the journal Drug Discovery Today. So in this article, we conduct a retrospective case study to design clinical trials with optimal p-values from the patient's perspective for hypothetical weight loss devices. All right, try and change the slide. And so in this analysis, we restrict ourselves to consider fixed sample, balanced, two-armed randomized clinical trials. And so this means that half the patients in the clinical trial are going to receive a control treatment of diet and exercise, and the other half of the patients are going to receive an investigational weight loss device. And so the optimization that we'd like to perform can be summarized by this figure below. So on the x-axis, we have our statistic, which summarizes the amount of clinical evidence contained within the trial that supports the hypothesis that the investigational device is more effective than the control treatment. And so in this example, if the statistic at the end of the clinical trial falls to the right of this critical line, which is our decision boundary, then the device will be approved. And if the statistic falls to the left of that critical line, then the device gets rejected. And so in this example, the critical line is set to 1.65, such that the probability of a type 1 error, the probability of a false approval, is set to 5%. This is also known as the statistical significance of the clinical trial. On the other hand, the probability of a type 2 error, the probability of a false rejection, is given by this area in yellow, beta. And so this is actually also equal to 1 minus the statistical power of the clinical trial. And so now we can use these patient preference scores to add another dimension, which indicates the severity of a type 2 and a type 1 error. And so in this example, here we see that the red color indicates that a type 1 error is more harmful to patients than a type 2 error. And now what our optimization can do is we can reset this critical line to a new value such that the expected harm to patients gets minimized. And so this will be equivalent to maximizing the patient value, the expected patient value. So to illustrate how we can quantify the severity of a type 1 versus a type 2 error using these patient preference scores, consider a device that has a % total body weight loss of 30% and a mortality risk of 1%. If we look at the patient preference scores for these attributes, we see that this device would provide 0.8 units of value to the average patient. On the other hand, consider a second device that has a % total body weight loss of 5%, but no mortality risk. If we again look at the patient preference scores for these attributes, we see that this device would provide patients, the average patient, with 0.2 units of average units of value. So we see in this case, if device B is received instead of device A, then our average patient would lose 0.6 units of value. And so looking at these relative differences between patient preference scores, we can see how we can quantify the harm caused by using an inferior treatment in the case of a type 1 or a type 2 error. And so in addition to the trade-off between type 1 and type 2 errors, our BDA framework considers another trade-off, which is the trade-off between the length of the clinical trial and the number of observations we collect, which ultimately will inform our overall error rate. And so to illustrate how we can quantify the harm caused by a lengthy clinical trial, consider a device that has a preference score of 1.0 and is available to patients today. Now consider a second situation where, again, we're looking at the same device, but now it's only going to become available to patients after a 1-year delay. Now, if patients discount benefits at a rate of 10% per year, then we see that the value of this device, the second device, to patients today gets reduced by 10% to 0.9 units of value. So in this case, we see that if there's a wait time of 1 year, then the average patient would lose 0.1 units of value. And so these discount rates, we can see how we can use these discount rates to quantify the harm caused by overly lengthy clinical trials. Okay, so then we can apply this patient value model, and we take this patient value model and we apply it to 4 representative sets of null and alternative hypotheses for our investigational weight loss device. So the null hypothesis in each one of these figures is given by the black circles, and the alternative hypothesis is given by the black triangles. Now, in each one of these subplots, the x-axis shows mortality risk, and the y-axis shows total body weight loss. The black contour line that divides each one of these subplots into 2 regions indicates different combinations of mortality risk and total body weight loss that have the same preference score as our control treatment, which in this case is diet and exercise. So any device that falls above this black line would be preferred to the control treatment, and any device that falls below the black line would be less preferred. And so again, we consider 4 different sets of alternative hypotheses for the investigational device. And in the case on the top left, we're considering the case of low mortality risk and high expected total body weight loss. And so this might be representative of a very promising, potentially breakthrough therapy. On the other hand, in the case on the top right, we see again under the alternative hypothesis, the device is expected to provide substantial benefits, but this comes at the greater expense of greater mortality risk. And so in this case, this might be representative of a device that requires an invasive surgery. Now, in the case in the bottom left, we have the case where the device has low mortality risk and low total body weight loss, and so this is a relatively safe device, but also one that's not expected to provide substantial benefits. And finally, in the case in the bottom right, the final case, we have the least optimal combination, which is high mortality risk and low total body weight loss. So actually, in this last case, we see that the alternative hypothesis is in fact less preferred to the control treatment. And so we'll see that our framework tells us that we should reject this device without even running a clinical trial. But in order to run the model for the other three cases, we're going to have to specify a number of parameters. In particular, we have to specify the size of the population that's going to be affected by this decision, the standard deviation of the response variable, the discount rate at which patients discount benefits, the patient accrual rate, as well as a number of other parameters that are going to affect the overall length of the clinical trial. We also apply a constraint to our optimization such that the statistical power of the clinical trial is capped at 80%. So we do this to keep our clinical trials in line with standard industry practices. Now, when we run the model with these parameters, we find that for the very promising, potentially breakthrough therapy, the optimal significance level is set to 6.5%, which is higher than the conventional level of 5%. This makes sense because in this case, the harm caused by a type 1 error of false approval, at least from the patient perspective, is relatively small. On the other hand, as we start to increase the risk of this device under the alternative hypothesis, we see that the optimal significance level drops to 1.1%. It drops to 1.1%. And so in this case, this makes sense because it makes sense to run a more conservative clinical trial in this scenario because we want to protect from the harm that would be caused by approving an ineffective device with high mortality risk. And finally, for the last case, where under the alternative hypothesis the device has high risk and low total body weight loss, we see that the framework tells us that we should reject this device without even running a clinical trial. Again, here, even if the device is slightly effective, the high mortality risk makes it less preferred than the control treatment of diet and exercise. But this doesn't necessarily have to be the case for all subgroups of patients. This is the case for the average patient. And so we can run our model again with different levels of risk aversion. And we see that as the risk tolerance of our patients increases, which is equivalent to their risk aversion decreasing, then the harm caused by a type 1 error, a false approval, actually goes down. And so these patients are going to be willing to accept higher rates of type 1 error, and in return, we see that the length of these clinical trials decrease. And so this provides these risk-tolerant early adopters with faster access to these new treatments. In fact, we also see in this case that in the case under the alternative hypothesis where the device has high risk and low total body weight loss, now our framework tells us that we can run a conservative clinical trial for our most risk-tolerant subgroup of patients. Okay, so to summarize what we've learned from this retrospective case study, we've seen that fixed significance levels don't necessarily maximize patient value. And in particular, for devices with low mortality risk, but potentially substantial benefits, and also for risk-tolerant subpopulations of patients, value gets lost by lengthy clinical trials that are too conservative and overprotective of the type 1 error rate. On the other hand, for devices that aren't expected to provide substantial benefits or might be expected to have higher levels of risk, then the traditional thresholds of 5% may be too permissive. And so in this case, the larger chance of approving an inferior device might actually not be justified by the expected benefits. So if you have any questions about this case study, we'll be happy to take them at the end. But at this point, I'd like to pass the presentation back over to Andrew for some concluding remarks.
Great, thanks very much, Joe. So where we are right now is we're collaborating with the FDA, Martin, Telba, and Murray, and working with RTI as well as the Michael J. Fox Foundation to apply this framework to Parkinson's. And we're really hoping to learn from that experience to see what aspects of our framework is practical and what aspects need to be changed in any way. And so we very much appreciate the opportunity to interact with those of you who are on the line and get your thoughts and hopefully be able to take some of these ideas and put them into practice sooner rather than later. So with that, let me conclude and thank Stephanie and MDIC and all of the participants on the call. We'd be happy to take questions or comments as you like.
Great, thank you. For those of you on the line, please send your questions in via the chat box, and it looks like we maybe have 1 or 2 questions already. I will look to pull those up. While we're waiting for those first questions, Sho, perhaps you can tell us a little bit about where the current status of this ongoing prospective project with the Michael J. Fox Foundation.
Sure. This is a really exciting project because we're actually designing a survey to understand what matters to patients, and we're designing the survey in particular so that we can apply it to this framework so we can design patient-centered clinical trials. And so currently we're at the stage where we've been working with patient scientists and councils and the Michael J. Fox Foundation to really try to understand the attributes that are important for patients who have this disease, and really trying to understand how we can quantify some of these different metrics so that we can use it within one of these risk-benefit frameworks. And we've gotten to the point now where we've actually designed the survey, and we are currently programming it, and we're hoping to start enrolling patients and inviting patients to provide us with their preferences in the very near future.
Yes. I just want to add that this is Martin Ho from the FDA. I just want to say that it's a privilege to work with colleagues from the MIT and Michael J. Fox Foundation. I want to emphasize the novelty of this project, especially it's the very first time that we are using designing our patient preference study from scratch in order to fully leverage the mechanism, the method that Andrew and Sho have just described to gain the most effective way for us to design clinical trials in a patient-centric way. Thank you.
Great, thank you. Well, we have a couple of questions that have come in. I'll put out the first one. I think this, Martin, this may go to you. Regarding the obesity study survey, how did FDA determine the levels for each attribute included in the weight loss DCE survey?
It's a very good question. That is a very evolved project that closely collaborated between the RTI Health Solutions and our review colleagues. So the process of determining the number of levels and also the appropriate level for us that would be of a regulatory interest is a very important process. It's a very important part of the process when we are designing the survey. So we take a look at the devices on the pipeline, and we look at what are the significant changes within each attribute that would make a difference. For when we are making regulatory decisions in terms of approval or not. So, and as for the number of attributes, the number of levels, since we wanted to maintain a reasonable level of power and precision of our estimate, we limited to sometimes within 3 levels. But for some important attributes such as mortality risk and other attributes, for those we are very interested to have a better resolution on the patient preference trade-off. We would assign more levels in order to get that precision of the information.
Terrific, thank you. I have a question here about the timing of evaluating patient preferences. Can anyone comment on the timing of evaluating patient preferences with respect to choices being hypothetical versus based on actual trial results? It appears the example requires a good guess prior to conducting the trial to assess safety and effectiveness.
It's also a very good question. I think based on the submissions that we received so far, I think the ideal timing of designing a patient preference study would be at a time when the sponsors have a pretty good idea about the range of the attributes that are important for regulatory decision. For example, although we don't necessarily have to have the precise point estimate of or the exact mortality risk or the exact amount of benefits that the device would deliver, as long as we have a range or a ballpark figure that the number where the clinical study result would land on, that would be a good time for the sponsor to come in to talk with the FDA. I would say that by doing that, we can first develop a consensus about what is important or relevant for regulatory decisions. And second, that will give us some time to develop a survey instrument that would be regarded as mutually regarded as neutral and biased and accurate. So once we reach the agreement, then we can conduct a patient preference studies. Despite we don't have the final study estimate in our pocket yet, we can still apply the study result to the clinical trial result when it becomes available. Otherwise, some people would also choose to do a patient preference study after they finish and have a very good idea about their about the clinical trial results. That can be done as well, so that it can be done in a way that the sample size or the range of the attribute level could be narrower than the previous approach. But I would say that the previous approach still would be a good way for us to go, mostly because it takes time for both the sponsors and the FDA to reach a conclusion as to what would be a good survey instrument to do. I think which is way more than just about the attribute levels and the attribute themselves. Great.
I have a statistics question here. This question is related to Bayesian versus Frequentist. It says that it seems that a Bayesian method is used to adjust Frequentist characteristics. Why not put patient preference directly into a Bayesian trial with appropriate weights, also given that Bayesian interpretation is more intuitive in terms of probability of certain outcomes?
For that, I would defer to Professor Lowe.
Oh, great. Thanks, Martin. So this is a very interesting point that was actually raised on a couple of different occasions when we were presenting results both to the FDA as well as to other organizations. So let me first start by pointing out that there's actually very little Bayesian about Bayesian decision analysis. And this was a point of contention with a particular Bayesian statistician who rightly made the point that if you're going to call it Bayesian, you may as well engage in some kind of adaptive design. And we actually have a paper that we're working on right now that does just that. That is, using the adaptive design framework that Don Berry and others have developed, we're actually incorporating differences in the costs of effectively type 1 versus type 2 error, although they don't use that frequentist notion, but basically making bad decisions. There are costs of that, and you can incorporate that directly into an adaptive design. In fact, I would argue that it's easier to do that in an adaptive design framework rather than to use a frequentist framework. The reason that we decided to start with the frequentist framework is that clinicians, as well as the biopharma companies, are currently still more comfortable with that kind of standard two-arm balanced randomized clinical trial framework, so that there are many, many more clinical trials out there that are being conducted in the standard way than there are using adaptive design. In fact, we actually ran into some skepticism at biopharma companies about the adaptive design framework. And clearly there are some very notable successes like iSpy and LungMap and so on, but the vast majority of clinical trials out there that are being conducted today are still being conducted in the classical statistical framework. So we wanted to use a framework that was not that different from what's currently being done, but different enough that it allowed individuals to take these ideas and start using them right away rather than to wait for the adaptive design framework. So that's the main answer. It's really out of practicality.
Great, thank you. I have a question about the values of this to manufacturers. The question is, how do you perceive the value to a manufacturer of a narrowly focused clinical trial and the higher risk tolerance patients? Does this limit the target patient population?
Can you repeat the question?
Sure. It's regarding the value to manufacturers of a narrowly focused clinical trial in higher risk tolerance patients. Does that ultimately limit the target patient population for a technology? It's
unclear to me what this question is trying to address, but I will do my best to answer that question. I don't think it's a limitation, rather it's a supplementary voluntary submissions of the, to help us to make a determination of the benefit risk profile of a device. So therefore, by providing this additional information, it will help the approval consideration rather than hurt the submission. So therefore, I would disagree with the statement that it will narrow the targeted or indicated population.
Great, thank you. Can you comment? I think in this we could probably talk about this with both the obesity study and the Parkinson's study about how your study teams or how our study teams chose the conditions that we studied.
It's a great question. And as for obesity studies, I think is widely known is the obesity situation is very prevalent in the United States. So we are very eager to deliver to promote good public health, and so therefore by studying the patient preferences, we can deliver a very good big public health impact. And on the top of that, when we are considering the patient preferences, when we are considering the weight loss devices submissions, we were facing a challenge that is also can be common or generalized to other situations, which is we are facing a situation where the device either can deliver a lot of benefit, but at the same time very risky, versus the other end of the spectrum is they deliver very safe treatment, but at the same time the effectiveness is minimal. So when something new devices or technology comes into our review, how can we determine such a benefit risk trade-off would be acceptable? So I think because of that, we chose the obesity study as our first patient preference study. And as for the Parkinson's disease, it has been also a very important public health concern. And we are also understanding that the fact that Parkinson's disease patients, they are very heterogeneous. They have different, they may be at different stages of their disease or conditions, and each stage would have been facing a very different treatment options regarding the benefit-risk trade-off. So therefore, just based on the public health impact and the importance of, and also the challenging, I would say, heterogeneous patient population among the Parkinson's disease patients, we chose it as another example for us to work on, so that we can learn from it and help us to make a better and more patient-centric benefit-risk assessment of the devices.
Great, thank you. After benchmark treatments are available, shouldn't new treatments be compared to standard treatments rather than no treatment? And this is referring to, as in the example of prostate cancer, without adjusting the p-value?
I would defer that to Professor Lowe.
Professor Lowe, are you still on?
Hi, I got a message from Andrew saying that his mic might have been muted somehow. So I'm not sure if he's able to reply right now.
It is not showing him as muted. Maybe if I.
Hello.
There we go.
Can you hear me?
Yes.
Yes.
For some reason, I don't know what's going on, but I was speaking before and must have gotten cut off. Let me answer very quickly before this goes out again. The answer is absolutely. As therapies change, you're going to want to change the appropriate weights for the optimization. And so you want to be rerunning the optimization on an ongoing basis and also taking into account patient preferences and learnings from clinical trials as well as other post-approval data in order to be recalibrating these kinds of weights for type 1 and type 2 error.
Great, thank you. Well, it looks like we have reached near the end of our hour here. We will be making video of this, the recording of this webinar available on the MDIC website. So we'll send you a link to that when that's available. You can also find additional upcoming webinars on the MDIC website, including another webinar on the topic of patient preferences that we have coming up on December 4th, talking about patient preferences in the regulatory review process. So we're looking forward to that as well. I want to thank Martin and Andrew and Shomesh for joining us today. This is an exciting and emerging topic in clinical trials and in medical device regulatory science, and we are excited to be able to share this ongoing work with all of you. Thank you very much.
Thank you.